I'm going to just give kind of an overview of the kinds of things that were discussed at Scientific Day. So this is actually only the second Scientific Day that FPWR has sponsored. We had one 2 years ago, and then we had our scientific meeting as part of IPSO last year. So we had about 70 participants, which was, I think, a really good sized crowd. It was maybe about 50 to 60 scientists. So it was enough that there was a critical mass of science, and they were really good scientists, but it allowed them to interact really nicely too. So sometimes you get to these ginormous meetings and you don't get to see anyone. So we had a few brave parents who came, and we had investment firms that came, which is something new for us and shows that the biotech investment community is trying to learn more about PWS, which was encouraging. So we had 16 oral presentations, 6 posters. Scientists, it's really like your 4th grade science fair. Scientists do posters, and we had a poster session. The vast majority of people who presented were FPWR-funded projects, although not all of them were. I thought it was very high-quality science, but I also got a lot of good feedback from the participants about how they enjoyed the science and felt like it was really good quality science. There was a lot of great discussion throughout the day as well, which was good. And it was even colder than it is now in this room. So it's very cold. I know. It was seriously, yeah. So I'm just going to go through the sections and tell you a little bit about them. We had, these are the 4 talks that were in the genetics and cell biology section. And so these are molecular biologists who are trying to tease through the complexity of the PWS region genes and see what happens at the molecular level when you lose those genes. So in the genes and then in cell systems. So let me tell you a little bit about that. So we've talked about the complexity of the PWS region. So this is the genes in blue are the important genes that are deleted or inactivated in all kids with PWS. And then this gene, MAGL2, is mutated in Schaff-Yang syndrome. And this is complex enough, but when you start, this is the DNA. When you start looking at the RNA, it gets super crazy, even compared to all other parts of the genome. There's all these crazy RNAs that are made, and then they're processed. So there's parts stuck together and other parts left out. And so some of the scientists study the parts that are stuck together, and some of the scientists study the parts that are cut out. And they're trying to decide what each of those things do and which ones are important for conveying the PWS symptoms. So which ones, when you lose them, you get PWS. And as you might imagine, it's a really complex question, and people are looking at it in different ways. So Stefan Stam has been looking at, I'll go back, he's been looking at the pieces that are cut out and further processed, and he's asking, those are little RNAs, and they probably go off and regulate other RNAs. So he's trying to figure out what are the other RNAs that are being regulated. And so he has found and reported on and is continuing to look at that those RNAs influence genes that are being spliced, and they modify what the end product of those genes are, number one. And the other thing that they modify is how stable the RNA is. So an RNA is made, and protein is made, protein is made, protein is made. If the RNA gets chewed up, you get less protein being made. And the SNORD 116 RNAs seem to be regulating a bunch of other RNAs and how stable they are, which is going to influence a bunch of other proteins downstream. So finding those targets and understanding what proteins are being influenced by our RNAs is really important, because if we can identify those proteins, we might be able to develop a therapy that doesn't act on the PWS region, but acts on the downstream proteins that are affected. And so Janine LaSalle is an investigator at UC Davis, and she is looking at, in the big picture, how the RNA changes over time, and she's found this circadian rhythm to the SNORD 116 RNAs. So at certain times of the day, there's lots of them, and then there's less of them. So they have a 24-hour cycle. And when you lose that cycle, when you lose the region, it influences a bunch of other genes that are supposed to cycle on a 24-hour basis. And so she believes that this is underlying some of the reason why our kids seem to lose that 24-hour cycle, and they're up at 4 o'clock every morning and they're sleeping during the day. That the SNORD116 is kind of a master regulator of a bunch of genes that are important in cycling. So this, at this point doesn't necessarily immediately point to a therapy, but it does say that something to restore the cycling, the 24-hour circadian rhythm would be important for our kids. And then a third project that was presented was a project that was actually published earlier this year, and they're continuing on. This is the group from Duke who did we know that every person with PWS has the PWS region there on their maternal chromosome, right? So the normal situation is you have dad's chromosome, excuse me, and mom's chromosome, and the genes are expressed or turned on the dad's chromosome. In deletion, the dad's chromosome's got that chunk missing. In uniparental disomy, there's only mom's chromosome. But the DNA is sitting there. It's there and it's there. It's just turned off. So Dr. Zhang's been using small molecules. I did a big screen of thousands of molecules to turn those genes on. He found that he can turn them on in cells, and then he gave them to mice. And so this is a survival curve. These are mice and how many are alive. So that he uses a very severe model of PWS mice, and they all die within the first couple of weeks. These are wild-type mice. They all live, 100, or almost all of them live. When you give this drug, it takes the PWS mouse and improves their survival. So he showed that his drug turns the gene on, and it has an effect in whole animals, which is a big step. It shows that, this is a potentially feasible therapy. The downside of his small molecule, and he is, with FPWR support, he's moving that forward to do safety studies, but it's not super specific. It turns a lot of other genes on, and that may or may not be a good thing. So he's also in his lab, he's collaborating with a guy who does a lot of, uses CRISPR to turn genes on and off. We most often hear CRISPR to do gene editing, but you can also use it to turn genes on and off, and it might be a more specific way to turn the PWS genes on. So they're moving this molecule forward, and then they're working on the CRISPR technology as well. And then one of the genes in the region that we know is really important is MAGL2, because if you just have a mutation in MAGL2, then you get Schaff-Yang syndrome, which has many of the symptoms we heard from Dr. Schaff yesterday of PWS. So really understanding what that protein does. So that's a relatively simple. It's a DNA, and it makes an RNA, and it makes a protein. And so two investigators, Dr. Potts and Dr. Weverick, have worked on MAGL2 and what it does, and they presented a lot of data in cell systems looking at what MAGL2 normally does. And this is really the first time, not the first time, I guess at IPSO, that we really are starting to see cellular phenotypes. So things in a cell that you can say, oh, this is different in a PWS or a Schaff-Yang cell compared to a normal cell. And that's a really important thing, because if you want to do drug screening, you have to be able to tell the difference between the two. So what Dr. Potts has really provided evidence for and continues to pursue is this idea that the MAGL2 protein is really important in moving. There's vesicles inside the cell, so you can think of like a cargo, trains inside the cell that moves proteins from one place to another and up to the cell membrane and back, and recycles the receptors that the leptin receptor and the ghrelin receptor and all the receptors that receive the signals from outside. And the MAGL2 protein is important in that trafficking process. It helps move those receptors around. So when you lose MAGL2 or mutate MAGL2, they don't move as well, and the receptors don't recycle as well. So Dr. Potts's studies are looking at endosomal trafficking. So again, the receptors come in and then they can get degraded or they can go back out. And that's where MAGL2 seems to be important. And Dr. Waverick looked at a specific leptin receptor which receives the signal from the fat cells that there's enough fat in the body. And she showed that the MAGL2, when you look at the receptor, you can see that it's not trafficking appropriately. So that's important because there's probably other receptors that aren't trafficking appropriately too. And that is something that potentially you could do a drug screen on and see what drugs would get those receptors to recycle more normally. You guys can, like, if you have questions or something and you want to, yes?
Do you think that the other possibilities that aren't trafficking correctly, do you think that's causing specific symptoms that we do need to check it?
Right, so the consequence of the leptin receptor not recycling so well is that the body doesn't receive the leptin signaling the way it should, which is pretty obvious because fat cells produce leptin, and so, and the role of leptin is to make you eat less. Kids with PWS who are overweight produce a lot of leptin, just like you would expect, but their brain doesn't receive the signal. And so this might be the reason that the brain isn't receiving the signal, because the receptors are going inside the cell and then they're getting degraded, or they're not getting back out the way they should, so the signal is not being processed the way it should. So yeah, so you could see, like, you know, you could envision drugs to get the receptors trafficking the right way, or you could envision drugs to overcome that leptin resistance in another way. Okay. Yeah, and there's the whole complex with MAGL2, and that's what Dr. Potts is really sorting out, is what proteins it sits with to carry on these processes. We then moved on to hyperphagia and endocrinology, and there were 3 talks in this section, and I'll just go through them briefly. The first one was a group in Cambridge, and the problem, we have many different mouse models of PWS, and the SNORD116 model, deletion model, is one of them. And the problem with the model is it doesn't get overweight, right? So you delete SNORD116, which is deleted in our kids, and they have the failure to thrive, so they're little mice, and they don't grow as well as they should, but they never get overweight. So you can't test obesity drugs in that, right? So that group is looking at a way to keep the SNORD116 genes there when their mice are born and for the first couple of weeks, and then delete it and see if that helps the mice get obese. And they've shown in a percent, they can get a percent of the mice to do that, and they're working on techniques to improve that. And so that'll be a really important model for the community, to have a SNORD116 mouse that gets obese so we can be testing these drugs in them. Dr. Zygmunt, who's talked to our group before and is a really good physician scientist at UT Southwestern, has an interest in ghrelin, and we know ghrelin is high in PWS, and we know that when people get a lot of ghrelin, or mice get a lot of ghrelin, they gain weight. It drives you to eat. And so, 2 of the clinical trials yesterday were to bring that ghrelin level down. But his lab has taken a different tact and done a lot of really good studies showing that ghrelin actually has some protective effects, particularly, and his hypothesis is that in the neonatal period, like after the babies are born, that ghrelin is actually being protective, because ghrelin also regulates blood sugar, and we know that a lot of kids with in the early neonatal stage have hypoglycemia, so their blood sugars go down sort of in an abnormal way. And so he is looking at experimentally with mouse models, is looking at what the role of ghrelin is in protecting newborn PWS mice from drops in blood sugar. And he actually is thinking that in young kids, babies with PWS, we might want to give them ghrelin. So he's doing that in the mice to see if that's a way to go. And Dr. Nichols has looked for a long time, that's this one down here, at the pancreatic cells in mice with PWS. And he's shown that in the mice with PWS, they don't organize the way they're supposed to be organized. And they don't secrete all the things that pancreatic cells, insulin, I don't know what else pancreatic cells, all the things that they secrete. They don't secrete them appropriately. And so he's doing cellular models of this, looking at defects in secretion, because that's another thing that might be defective in PWS. And he's going to use those, again, to understand the basic defect and potentially to do drug screening. So we then moved on to neurobiology, which is complex. And one of the things I'm really excited about, though, is, you know, like 4 or 5 years ago, we had very few neurobiologists, which, like, how crazy is that? I mean, you would think all the neurobiologists would want to study PWS, but they really didn't. And so we kind of tried to be aggressive about seeking them out and making sure they knew about our funding, making sure that they knew that PWS was an interesting disorder to fund. And we now have several neurobiologists who are interested in PWS. So I think that's a wonderful thing, because I think we really need to look at how the connections in the brain are different in PWS than in typically developing kids. And so, I'm not going to go into great detail, but Dr. Kurash from University of Calgary came, and she was talking about these immune cells that normally are in the brain and that increase with obesity. We know that. This is a picture from her of the brain, and you know, which includes these inflammatory cells. And she's looking at, in the MAGEL2 mice, what those cells are doing, and she sees an increase in them, and, you know, what the consequence of that is. So it's known that whole inflammatory state in the brain can aggravate obesity, and so she thinks that's probably a component of what we see in PWS. Dr. Tucci is looking at specific sets of neurons, the MCH and the orexin neurons in PWS, and looking at how those impact. So orexin neurons are known to, MCH neurons influence appetite, and orexin neurons influence sleep, and how those interact, and in mouse models of PWS, and how they underlie some of the appetite and the sleep disorder. Actually, this was Larry Reiter actually presented, that's his graduate student, a study using the teeth that you know, people have given, and asking of the cells that have come out of them, how are they different than normal cells? And this is I don't usually show primary data, but like here's what he showed. Like, anybody could, like, you know, those are the gene networks that are impacted by loss of the PWS genes. So it just shows you it's a complex thing, but it also shows that there's bioinformatics that can be used to organize that and look at pathways that are being disrupted in the dental pulp stem cells that he's studying. So again, that's looking for what are the downstream consequences when the PWS genes are lost, and are there genes downstream that we can be targeting? And finally, in the neurobiology section, the group from this Mount Sinai School of Medicine is looking in donated autopsy brains from individuals with PWS, and they look at a particular set of neurons, the von Economo neurons, which apparently are only present in primates and higher, so monkeys and higher, and really important in social interaction. And they found that these neurons are missing, or they're not oriented correctly a lot in autism, so they were interested to look in PWS. And so far, interestingly, where they expected to see less neurons, they're seeing more neurons, so that was surprising to them. But they're going back and looking at different parts of the brain to see if the neurons are maybe just sort of in a different place than, you know, spatially different than in the typical situation. And finally, there was a session on the clinical genetics, behavior, and mental health. And Virginia Kimonis, from who some of, or maybe many of you know, from University of California, Irvine, she was part of the NIH-funded a rare disease network that included the Jen Miller site in Florida, Dr. Butler's site in Kansas, and Dr. Kimonis' site in California. For many years, that funding ran ran out about 2 years ago, but they collected data from my child and many of us who have slightly older kids, and have been looking at all of the data, the clinical data. So she reported on the sort of the birth period of that data, you know, how long kids had NG tubes, and you know, how long it took to get the diagnosis, and you know, all of those features around the birth time. And that data will be great historic data for our registry, for the data we're gathering now and will continue to gather moving forward, to see how these practices around the neonatal period are changing over time. Dr. Dorenberg is with Anastasia Dimitropoulos at Case Western, and she is doing this PRETEND study that FPWR is funding, which is a telehealth intervention where they work with families to encourage play in children. So play is a really important tool by which children learn social interaction. And so the goal of this program is to coach parents onto how to help we know that kids with PWS don't play the way typical kids do. They play less, and they're not as elaborate in their play. So it's teaching parents, coaching parents on how to help their kids develop. Because what they found is that the kids, if you leave them like, you know, with a couple of stuffed animals and some blocks, they don't play very well. But if you come in and you give them some cues, they're actually pretty good at it. And that's actually different than kids with autism, or they see differences, that kids with PWS pick that up a lot quicker, so they really feel like there's a chance for intervening. So they're coaching parents on how to you know, work with the kids and get them playing in a more typical manner. So they're in the middle of that study. You may see we've been advertising it online. And so they didn't report any, so they're doing this intervention, and then they're going to see how it impacts play. So they didn't report the end product of that. They really reported their initial screening, which showed this deficiency in play and sort of described the aspects of that. Lauren presented work that she and Natalie have done looking at the high levels of caregiver burden in PWS. We all know that it's very stressful to have a kid with PWS. Really right from the beginning, from like the time you're dragged into the NICU, which was not in your birth plan, all the way through them, you know, being adults. Like we all knew that, right? But documenting that is really important too, again, to provide evidence to drug companies and to the FDA that there's a real need here, and it encourages drug companies to see that data, so they know that when they go to the FDA, they're going to, you know, essentially be taken seriously. It's good for the FDA to have that data so that they can really understand that there's a need for drugs here. So again, a little plug, because we'll be doing a follow-up to this study looking at caregiver burden over time. We'll be doing that through the registry. We'll be linking it to the hyperphagia data. So if your child has more behavior around food, is it more stressful? We think it probably will be, but we again want to document that and see how that interacts. And Lauren and Natalie will be doing a webinar this fall, kind of going through that data and talking about maybe ways, now that we know we're going to be stressed as parents, so you know, what can we do to you know, help ourselves. And finally, Tony Holland came from Cambridge, so we had 2 people from Cambridge, and talked about his study using transcutaneous vagus nerve stimulation in PWS. So some of you may have heard of VNS, and people have looked at it for obesity. And so a few years back, Tony said, well, let's take a couple of PWS patients and do the VNS and see if they lose weight. And so they just took 3 patients, because at the time it was a surgical procedure, so they had to implant these things. So they could only get ethical approval for 3 individuals. And it turns out those individuals did not lose weight, but their behavior changed in a big way. So 2 of the 3 had very aggressive behaviors, and the behaviors, the parents said, just sort of like melted away. And you know, when the study was supposed to be over and they were supposed to turn the VNS off, the parents were like, no, you got to keep that on. And one of the young women went from, having a lot of bad behavior to she's actually married and living independently now. She has a husband who has a Williams syndrome, and they have supports, but they're living independently as a married couple. And yes.
So, like you were just saying, the mom said, like, please don't stop. Is this something that is expected to have lasting effects once you stop, or it would be an ongoing sort of treatment?
It would be an ongoing treatment. And someone asked Tony that. He said he would expect that you would get the behaviors coming back pretty quickly after, yeah.
And it would be, I think I actually reviewed this study, but I don't remember. So how often would it be?
So what they have now is a transcutaneous one. So it sits on the outside, and I guess it's got some probes I guess they sit behind your ear. And so it's not a surgical.
Continuous stimulation?
It's continuous. Well, they do 4 hours a day. 4 hours a day. But I guess you get used to it and you don't really notice that it's happening. So they're now doing, Tony's a very excellent, careful researcher. And so they took 6 months of data, baseline data, really monitoring how many aggressive acts per day. Because the issue with the first study was they didn't measure changes in behavior because they didn't expect changes in behavior. So this study, they're really focusing on the behavioral aspects. And so they have 6 months of data really documenting the aggressive behaviors. So they picked the people who had the most aggressive behaviors, which, as you might imagine, is a challenging group to work with. So it's this thing, like you want to hit the people who have the most need, but those are the people who have aggressive behaviors. So sometimes the VNS went across the room because it's not implanted now. So and now they've turned the VNS on and they're gathering data too. Apparently it takes a little while to see an effect. It takes probably, I think he was saying 6 to 8 weeks. 4 to 6 months, yeah. So they're at that stage where they're gathering that data, and so that was interesting. So I told Tony, like, if it works, so we're flying to England, yeah.
Yeah, so let's say it works. Like, how does the approval process from the FDA for medical devices? It's not.
Actually, medical devices tends to be a lot more responsive to patient preferences. They have sort of led the way on the whole incorporating the patient viewpoint, and they tend to be as long as it doesn't hurt you, and I don't want to speak for the FDA, especially since we're being recorded. So, but they have a lot of flexibility. Like if patients, as long as they feel like that it's a safe treatment. And I think VNS is approved for some indication. Okay, yeah. So presumably, it would be pretty accessible fairly early if we can show that it works. And the other nice thing about the transcutaneous is it's not surgical, so it's much less expensive. And of course reduce risk. But the question he has is, does it work as well as an implanted device? Because it may be that the implanted device works better, which would be a pain.
And the 4 to 6 months is based on the epilepsy study.
That's, yes. Yeah. And then finally, there were several posters. Rachel Weverick looked at the drug that Selena is using, the diazoxide, the clinical trial. She looked at that in the MAGL2 mice and showed the MAGL2 mice do become obese eventually in adulthood. Not hugely obese, but they have increased fat mass. And she showed that the diazoxide works in the PWS mouse model, which is nice supportive data that suggests that it is promising in PWS. Jessica had a poster on the registry. She's going to talk more about that later. Everybody needs to join the registry and fill out their surveys, please. But she's got some good data from that. And Walter Tucci, who came from Italy, presented some of his other data on using pitolisant in a mouse model of PWS, and also just looking at other. He's a sleep kind of guy, so he looks at all aspects of sleep in PWS. Natalie presented on the preclinical animal network that FPWR is supporting. She'll talk about that some later. And then the group from Kentucky also talked about how loss of the SNORD115 genes impacts serotonin biology in PWS. So that was our poster sessions. So that was an overview. Oh, and it's 9:15 now, so you can still join your breakfast groups if you want, or I'm happy to talk about any of the projects or things that were presented. Oh, yeah, so all of our grants are reviewed by both scientists and then advocates. So families, tell us if the study is relevant to them, and the scientists essentially tell us if this study is scientifically sound, and then we prioritize based on both things. So they have to be, good science that's important to our community. And so we're always looking for advocate reviewers to help in that review process. Yes, ma'am.
What kind of age range do you have in advocate?
We try to span, and because some grants on behaviors, obviously if a parent is older, they understand that better. So we try to get advocate reviewers across the board. So Jessica has a list. If you want to send your name to Jessica, she'll. Yeah, no, we always need more parents of older kids because we have lots of the younger, and so especially if you have an older child, it'd be great for you to.
How do you define older?
Older, I would say over 8, yeah. So any other questions?